So I'm trying to talk about our experience, our developments. I mean, these are developments, so be aware it's not a final product, something you can buy off shelf, but you can already try things out. And there's a lot going on and a lot development. And I'll try to explain why we're developing it and what's the ratio behind it and what's the concept. So let's just start a little bit with how modern chemistry is developing. So just a few use cases. So maybe one is heterogeneous catalysis. So you have like hydrogenation of carbon dioxide. You want to build research on that, build reactors, build reactions, understand this, study these things. For these to study, it's not only required to just draw the chemical reaction or reaction scheme and think about it a little bit. It's much more, you need to understand the materials that you are using in this heterogeneous catalysis. You need to understand, build devices, very complex devices, flow chemistry, reactors. You need to simulate the reactions. You need to analyze the products, do maybe even machine learning with it. And maybe at some stage also sell this product. I mean, maybe you build methanol and you can build it as a large factory. So you need a lot of information come together to keep and store and make available to do this research project. Another example is bioplastics. So the lactic acid polymerization. So here's the very basic scheme. To do this, you need to understand or find the right source from bioresources like the starch from mice or whatever, and then you need to ferment that to lactic acid. You need organisms. You need to understand how the fermentation process works. You need a fermentation reactor. Then you need to isolate these products, and then you need to polymerize them under certain conditions. So you need to understand the polymerization process. And then finally, you get some products, and you need to characterize these products. So you need devices and measurement types for these products. And maybe you also want a 3D printer running with a PLA, and then you need to even study the material properties there for the print process, the printed product in the end. So a lot of information you want to store, except of the pure chemistry around it. A third example is sitagliptin synthesis. So if you want a drug, you synthesize a drug, and so for example, you can so modern synthesis routes in pharmaceutical industry, they try to replace many of the normal, the organic or inorganic synthesis steps by biocatalytic steps. Here's a very interesting example where you have a very selective, see here, this amidation from this ketone to this amine with a transaminase. So in the yield, you see it's very high yield and high enhancer selectivity. And these things you can't easily do with normal organic chemistry, or it's a lot more complex. So biocatalysts have especially won quite some field in this large scale synthesis. And to develop such biocatalysts, such enzymes, you need a lot of knowledge about first the organisms which can be used to where you find such transaminases, for example. then you need to express these transaminases. You need an organism, an expression host. You need DNA that you need to modify it, because the transaminases in nature, they don't transform this pro-sitcaliptine into the final product. They do something completely different. So you need knowledge and know-how about the DNA, is the gene building methods, so the molecular biology. You need to build mutants. You need to characterize these mutants, make some transformations and put them into the cells and express them, screen them. For that, you need, for example, such a robotic platform like we have it here, the LARA platform, or a microfluidic system, or whatever you need. You need an assay that shows that this conversion really happens. You need then to isolate the product, so all this downstream processing. And then you need to make, of course, a medical product out of it. So you need to have all these clinical studies. So and a lot of data needs to come together to study such things. So what I want to say is that a simple electronic notebook system like Chemotion or something like this is not helpful, or is only it's helpful, yes, but it only can cover like a very minor fraction of all this knowledge. So what we want to achieve is covering everything. So covering, or not everything. I mean, that's a little bit too bold, but at least as much as possible, and especially the things that are the information that is required, first of all, to reproduce the experiments as much as possible, and also to learn from these experiments and share them in a fair way. So what we want to achieve is that we plan this experiments and execute them and capture all the data and metadata and make the data findable, accessible, evaluate the data, share it and publishing it, and then get feedback and refine it. So you have this complete build, test cycle, explore cycle, modify cycle. So it's a very simple idea what you want to achieve. So everyone wants to have this thing. So it's a very clear thing. But if you look into the details, you realize that doing such things, building such a thing is not easy. You need a lot of detailed information. And if you know a little bit about rocket science and rocket building, then you know that all these small little lines here are not parts itself. They consist of many parts. So it's not the final thing. So they itself consists of hundreds and thousands of small parts, screws, transistors, etc. So it's a lot of work, we realized, but at the beginning we thought it's a very simple thing. And what we came up with is currently this LARA architecture, which I tried to explain. And it starts with so it's a modular system. So you can't have like a monolithic system for such a complex thing. You need like a distributed system with a lot of modules, and you need to be able to choose what kind of things you need for your research. So we have basic modules for experimental planning, for the process design, for the materials, to define the material, to store the materials, like a report inventory of materials and of substances. And you need, if you want a biological study biological things, you also need information about the organisms. So also all of that is in a module that defines the experiments. And then you do the experiments. And the experiments, if you want to do them reproducible, you need to specify them in a way that can be read or reproduced by many others. And for that, we developed a language. It's Python based, but it's not Python. So there's a subtle difference in it. This language is so generic that it can be used to describe all kinds of things. So it can be used to describe chemical reactions. It can be used to describe processes in robotic platforms. It can be described to do some all kinds of everything that is somehow serializable, put into or can be expressed by a stepwise protocol that can be used, transformed into PythonLab. And then we have an orchestrator, and this orchestrator and scheduler, these modules, again, some modules, they then orchestrate the things so that they talk with the devices and with algorithms, machine learning algorithms, for example, or even to humans and get feedback. And for that, we use this open SILA standard. It's a standardization lab automation communication protocol. And with that, we talk with these things and get data from them again. So we can, for example, run a certain process. And it's not limited to a robotic platform or fully automated system. You can also run a reaction, a chemical reaction, fully manually. It's just that you document all your steps and get feedback and put feedback of your steps or changes of your steps back into the system, that all the experiments you do are documented. And once some data is produced, we have a data collector microservice, and that gets notified and then collects from the sources that produce data and put them into databases. And for that, we have two different kinds of databases. One is just a very classic relational database, PostgreSQL in our case, that stores tabular data. So, for example, a lot of the substances is very suitable for, so millions or hundreds of thousands of substances you could store, sequences, organisms, or whatever. That can be stored in tabular databases, relational databases, and then the semantics, so the knowledge graphs, so the knowledge about the metadata about the experiments, that is better stored in a triple store, in our case, it's Virtuoso. And with this data, we also have built synchronized exchange data modules so that you can exchange this data to others and then visualize this data, query this data, evaluate this data, and use it to even feed machine learning algorithms into it and then learn from the data. And that's a very important thing, that this data it's very structured and gets semantically annotated by automatically. So you don't, as a chemist, as someone standing in the lab, you don't want to do all this stuff on your own by hand. So what we build up is an infrastructure that helps to automate a lot of what is usually required to be entered into the lab notebook. So we can really focus on the core things. So maybe just a few ideas. So here's one example of this language that we developed, this PythonLab language. So you can almost understand what's happening. So you move something, then you incubate something, then you move again, then you do a measurement, and then you move again a container in different locations. And if you are a little bit familiar with programming language, you will immediately grasp what's happening here. So it's a very simple notation, and it's very flexible. So how we process this, that I already explained, so you can skip it. And here's an example of how complex that can be. So with a few lines of code, you can generate like screening processes for our robotic platform, for example, with a lot of conditional things and decisions and loops, whatever you need to describe a process. Okay. Yeah, I mentioned the SILA standard. If you have questions to that, I can answer that. So what kind of data do we want to store? We want to store the materials, the physics information, the substances, the reactions, the process, and the organisms. So this is at least the core of the things that you want to store in a database. And for that, we developed this LARA database, which covers many of these aspects. So we have, as I mentioned, for data, for the materials, for the devices, for labware, for the samples, for the organisms, for the projects, for the processes, for the procedures, whatever you need to describe your experiment in a common life science or chemistry lab can be stored here. Okay, and then how does the data now come into the system? That's a very critical point. You don't want to enter this data. You don't want to have like proprietary data formats. So what we develop is a lab data reader. This is a Python module that converts information from a device, so files that are proprietary, into a standardized format. In our case, it's JSON-LD, and a special kind of JSON-LD we call SciDat, and I'll come to that in a moment, what this is. The information is very structured, so we use ontologies, so very clear, defined, well-defined term systems with the relations to each others to make the information also machine readable and findable, and make it understandable by machines. That's the most important thing. So readable is easy, but understandable is something more complex. And for that, we use EMMO as our main top and mid-level ontology. So if you have questions to that, I'll skip a little bit, go a little bit faster, because the reason why we use it's very well designed for fundamental chemistry research. So it has this very strong good representation of quantum physical entities, of particles, subatomic particles, of a good representation of physics in material. So it comes with all the basic terms that you need, like units, SI units, and things like that. So it's very useful, and it has also a Python representation of all of that. So you don't need to write your ontologies in these strange notations like turtle or even more worse, these XML representations. And we build up an ontology development pipeline, so we can constantly develop these ontologies. All of that is open source, and it's on GitLab. Just search for open source lab, and you will find our ontologies. So now it comes to SciDat. I mentioned already that SciDat is a current development to store data, scientific data, in an open source and very linked or semantically annotated format. It consists of two parts. So of the tabular data, and which is stored in a Parquet or packed as Parquet, and then you have the metadata, and this is stored in JSON-LD, and both are linked together in one file that can be stored very compact, very efficiently on hard drive, and then it can easily, very, very fastly put into memory, and you can very quickly load gigabytes of files within seconds. So this file format is used, fast Parquet format is used by data scientist people, so the people that have to crunch a lot of huge amounts of data. So that's from this data people. Yeah, and very importantly is to note is it has proper nullable data, missing data handling. It has a type for not available or not measured data, and has a proper nullable, and you can also specify constraints. You can say this data should be in the range of from 0 to 350. And if a machine learning algorithm sees that and sees a value that is above, then it knows that this data is not consistent. Things like that. So a lot of information can be stored in this SciDat format. Okay, I'm not doing that alone. So we're building that together with people from NFDI for CUT, for example, but it's expanding. So we also talk with NFDI for Chem and other NFDIs. So it's a growing thing, and we learn a lot from each other. What do we do with the data? So you want to structure the data in a way that you can search it. So we have a built-in SparkQL interface into the software. So it's Virtuoso is coming with it, so we can query the data, so do very complex query of the data. That's one of the aspects. But also, yeah, you can use machine Jupyter, your notebooks, to retrieve very easily the data. So we have a very simple interface to the data, and a system to node the data easily. So, or to name the data that you can, so good simple naming schemes that you can access the data. And of course, you want to do machine learning with the data, or you want to do some feedback system. So we have we're building infrastructure for machine learning of this data, to get automatic semantic retrieval of the data. I can explain that in the questions there part. So the infrastructure is designed that you can combine many of these instances and share data, and even finally, this is under development, put your data in Zenodo or the European Science Cloud, or even I'm also looking into the Chemotion database, for example, so that you can also upload to whatever. So since everything's open, you can, and it's a very let's say high information content, you can always move it to lower information sources that like aggregated data sources. Okay, we're building that on a very stable, robust software stack. So we are using mainly Python, Git, of course, GitLab, Docker, and Kubernetes will be used for the deployments, or are partially used for the deployment, Sea Lion Animal, and the EMMO ontology development. So there's a lot of software involved, but or it's different packages involved. But we try to choose software that is very robust and very well widely used and stable for long time, stable. That's very important. So with that, I want to close and just give some perspectives. So we try to make this as simple, this whole system, as possible. So we try to add like mobile web applications, mobile connectivity that you can use your tablet, your smartphone to enter data, to control your workflow, to get feedback into the system. These SparkQL queries are not very user-friendly. So we're also exploring large language models to build queries with that, which is not trivial. And we try to involve these ontologies that they get more consistent, and we can use these also for consistency checks in our data. Then that's the so intermediate goals that we try to achieve. So, and by that, I hope I could convince you or show you that we're building a very large community-driven open source infrastructure, and we try to really avoid black boxes. So everything that we are trying to use should be open source and should always be open. So also our products are open. And we hope that at some stage we can even build like a feedback system where a kind of robot scientist entity, like an artificial intelligence, can use this data and build and decide, build hypotheses and make feedback loops, and reuse the infrastructure to do new experiments that are creative. So that's something that we really look forward to. So by design it's possible. So all the infrastructure with the automation can be used to really build a robot scientist, basically. Yeah, and of course, machine learning, that's a more closer goal. We will try to apply this data to machine learning algorithms and use and see if we can really extract data automatically based on the semantics of it. That's something we will very soon explore. Okay, and we need, of course, more people. So if you are interested in that developments and like the ideas and want to explore a little bit how this all works and see it in action, then please join our development teams, or at least our meetings sometimes, and or just ask me, and I'm very happy to explain this very huge system in more detail. Yeah. So last but not least, I would like to thank a lot of people that are involved. So people from the TU Berlin, from my friend in Trondheim, in Lille, our group in Greifswald, and then there are teams like the NFDI for CAT, NFDI for Chem, I also should add here, and the Kiwi team that tries to really use this platform, and the sea, land, animal teams where I'm also involved for the development, and also our money providers. And with that, I would close and thank you for your attention.